Sienna's first product is based on the detection of the biomarker hTERT, which is a component of an extremely important enzyme called telomerase. The discovery of telomerase and its role in assisting cells to reproduce indefinitely was so impactful that it led to a Nobel Prize for the Aussie scientist and the team who discovered it. When telomerase is present in cells, it repairs the ends of chromosomes so that the natural wear and tear that happens each time a cell divides and reproduces doesn't shorten the life of the cell. That's a good thing when the cells are normal, highly proliferative cells in the body, such as white blood cells. We need them to proliferate, so we want telomerase in many of our body's cells. On the other hand, we do not expect to find telomerase in normal, non-proliferating cells, such as mature epithelial cells. These cells would normally just go through the usual cell aging cycle and eventually die off. It's been shown that 90% of cancers also use telomerase, and that's where it becomes a problem. Telomerase can allow the cancer to grow and spread without being subject to the limited life cycle of normal cells. Therefore, if telomerase is detected in certain cells where it's not expected, it can be an indication of something abnormal and may be associated with the presence of cancer. Sienna's test can identify the presence of hTERT in these cells through a special form of urine test performed in the pathology laboratory. Sienna's test can simply be added to the standard form of urine testing called urine cytology, which involves the pathologist analysing the size and shape of cells under a microscope. A positive test for hTERT in mature urothelial cells, which are the cells lining the bladder wall, is an unexpected finding and indicates an altered process of some kind. The finding of hTERT in these cells, when reported to the diagnosing physician, can provide a useful piece of information to assist in the diagnostic process. It means that there is a higher likelihood the patient either has cancer or pre-invasive urothelial proliferation, which may progress to cancer. A negative hTERT result can also provide a useful piece of information to the urologist. Urine cytology is well known to have low sensitivity for detecting cancer. This is particularly true for early stage, low-grade cancer, where the size and shape of the cells may still look quite normal. Telomerase, however, is upregulated even in low-grade disease. Since 90% of bladder cancers upregulate telomerase, then a negative hTERT result in support of a negative urine cytology indicates a lower likelihood of urothelial cell carcinoma being present than with a negative urine cytology result alone. The team at Sienna is also working on uses for the test in other sample types, where we hope it might provide equally useful information to assist the diagnostic process for additional cancer types besides bladder cancer. The addition of hTERT testing as an adjunct to urine cytology provides useful clinical information to better inform the diagnosing physician about the likelihood of urothelial carcinoma being present or developing.